right, well, welcome to Crops TV. My name is Darren Mueller. I'm an extension plant pathologist here at Iowa State University, and today's talk is going to be around soybean disease management. So the general outline, it's a little bit of a hodgepodge, but we're going to go and dive into a little bit to the literature and look at sort of some updates on Diaporthe and Phytophthora, both sort of in neighboring states. We've had several different papers that have been published giving some insight into both of these diseases or disease complexes, and then go over sudden death syndrome, risk management and risk update, and then also look at some product evaluations for both foliar fungicides and then SDS seed treatments. All right, so let's just dive in right away for Diaporthe. This is a complex of fungi that can cause several different diseases on soybean. The first one is Phomopsis seed decay. This has been renamed to Diaporthe seed decay. So from here on out, we're going to refer to it as Diaporthe seed decay. And also stem canker, both northern and southern stem canker, and then pod and stem blight. So what you see in the images here, you have pod and stem blight as the top image, and then stem canker as that bottom image. Again, Diaporthe, different species of these can cause these different diseases. We'll first focus on Diaporthe seed decay. This is a disease that will show up, especially when it's warm and wet later in the season or during the reproductive stages, R3 to R7, but especially as you get later into the reproductive stages. It's also more common in early maturing soybeans, and then also if harvest is delayed. So both of those are factors. So the early maturing ones would be that it's maturing when the temperature is a little bit warmer, and then the delayed harvest just gives you an increased chance of having the ideal weather for this disease to really set in. So the paper that we're going to be discussing is published out of South Dakota State, and Fabina Matthew is the corresponding author for that paper. So what they found in this paper was actually we typically consider Diaporthe seed decay to be caused by Diaporthe longicola, or Phomopsis longicola, now been renamed to Diaporthe longicola. But what they found actually as they started diving in and looking at the complex of different fungi that could actually cause these symptoms, they found 11 different Diaporthe species that can cause seed decay. And you can see some of them in the image here. And so some of these aren't actually pathogens that cause the pod and stem blight and stem canker, but other ones are new. And so, and in general, they all have that same white fluffy growth. Some of them are a little bit whiter or fluffier than others, but they have similar symptoms. Now, since the publication, there's actually been a 12th one, Diaporthe novum, has been added to the list, and the Diaporthe novum causes very similar symptoms as the longicola. So you can see that's the one that sort of is the fluffiest, I guess, white growth that you see in that top left part of that image. So the challenge is really all of like sort of the first question is does this matter? And I think so. While all of these cause very similar symptoms, knowing what the actual pathogen is or the species is really going to be helpful when we start breeding for resistance or looking at fungicide resistance issues or things like that. And so diving in and really trying to figure out what the pathogen is and getting the right species is a very important thing. And knowing now that it's way more complicated than we originally thought, I think that this gives some credence to the fact that more research needs to be done to make sure that we don't have, like, all of these species do behave differently when we're trying to manage them. And so we have to sort of take into account for that. Now, just a quick summary, just a one-slide summary, some of the research that was funded by United Soybean Board. We looked at, after Diaporthe seed decay, there was an outbreak of it in 2018, and so we had a graduate student, Alex Hafke, who looked at different seed treatments in both low and high quality seed. And so we received seed that was highly infected with Diaporthe, and we had a similar seed lot that did not have Diaporthe. And then we looked at different management strategies. So in general, the seed treatments did increase yield for the low quality seed, but not the high quality seed. the seed treatments did increase field emergence for both the low and the high quality seed. The one, I think the most interesting study that Alex did was looking at, does an infected seed sort of have a long-term effects on the vigor or the health of that plant. And so we did a study where we looked at both low quality and high quality seed. We established them and planted them at different populations and then thinned them down to the same population. And so the stand establishment was, we artificially made it the same. And then what we found was that low quality seed, it did affect germination, but once you had that stand establishment, we didn't see any differences between the low quality or the high quality seeds as far as vigor, canopy closure, and ultimately yield. And so there didn't appear to be any further ongoing issues from that low quality seed. It was just affecting germination, and then after that there was no effect. All right, so Diaporthe seed decay and how do foliar fungicides affect it? There was a study done by Fabina Matthew up at, she's now at North Dakota State, but she was at South Dakota State when the study was done. And so what they did was they collected pods from plots that had been sprayed in 2021 and 2022 with 3 different fungicides, and then there was a non-treated control. The 3 fungicides were all in different classes. And so a QOI or strobilurin was headline, the SDHIOs, Endura or Boscaline, and then the DMI or the triazole was Folicure. And these were all sprayed at R3. I'm not even going to show the data, but essentially what she found was that these foliar fungicides did not affect Diaporthe, causing seed decay. Now, if you dive into the literature and go back into the 80s, and even more recently, I think there was a paper that was done maybe 10, 15 years ago now here at Iowa State. If you spray the right product and at the right timing, usually a little bit later than R3, sometimes you can see some decreases in Diaporthe seed decay, but it wasn't very consistent. So this does match in general what we find in the literature, although there are exceptions showing that fungicides may work under certain conditions. So let's now talk about some of the other diseases. So we have seen stem canker and pod and stem blight. They've been more prevalent the last couple of years. And people, I get this question a lot. We're like, why are we seeing so much of it? I would say that there's a couple of things. Both of these diseases I would call stress diseases, where they're going to start showing up when that plant is experiencing some kind of stress. And we have seen plenty of stress the last couple of years. If it's too much water, too little water, temperature extremes, etc. And but I'd say probably the thing that's driving it the most is that we have some cultivars right now that are more susceptible to both of these diseases. And so that's going to really drive what shows up out in the field is the genetics. And right now we just happen to have some varieties or cultivars out there that are more susceptible, not just to the stem canker and pod and stem blight, but also to brown stem rot, SDS, white mold, etc. So, and so that's I would say those are the 2 main factors. There's stress, and then also there's some very susceptible lines. Now, one of the questions that I've been getting is, do foliar fungicides somehow relieve the stress? And so I'm going to dive back into, this is actually a study that was done in South Dakota, again by Fabina, looking at an outbreak of pod and stem blight in one of her foliar fungicide trials that was not targeting pod and stem blight. And so this is sort of just fortuitous data. And you can see the yield responses yields on the in the graph there, and they're all very low. But more importantly for this is that the fungicides did not really protect any of that yield from that loss from pod and stem blight. And I would say this is not a surprise. I would say when you look at some of our data, we didn't look at very specifically at pod and stem blight, but we had a study back in 2000, I don't know, probably about 10 years ago now, where we looked at late season stem diseases, and the foliar fungicides in that study did not affect late season stem diseases either. Well, even if the fungicides are not really affecting pod and stem blight or stem canker out in the field, or even consistently affecting it on the seed, one of the things that we are trying to keep an eye on is are we starting to select for fungicide resistance? We know Diaporthe is in every field all the time, and so when we do spray a fungicide, we could be selecting for some kind of sensitivity issues with different fungicides. And so this is a study that was initiated. We're collecting isolates from several different states and sending them up to now North Dakota. We're in Fabina's lab, and right now we have isolates of Calavera, Asclepiae, and Longicola, and looking at how sensitive they are to the QOIs, or azoxystrobin is what we're using, the strobilurins. And essentially what we're finding is that we are seeing some of the isolates within all three of these species are having some differences in sensitivity. And so we don't know if this is, if there's actually a genetic mutation that's occurred to cause a sensitivity issue, or if they're just naturally resistant or tolerant to the fungicides. And so we're continuing. Some of these isolates are now being shipped to Southern Illinois, and we're looking for mutations and trying to confirm if it's actually fungicide resistance or if it's something else. And so stay tuned on that, but that hopefully we have better news there. All right. So that was Diaporthe. Again, a sort of a complex of several different diseases, and now we know that there's a lot of different pathogens that are sort of mixed in to those diseases. We're going to move on to Phytophthora root rot, and actually in the last year or so, year and a few months, we've actually had 3 papers being published. One that was out of Michigan State and Marty Chilver's lab, one of them out of South Dakota in Emmanuel's lab, and then actually one here at Iowa State in Allison Robertson's lab, and she's working with colleagues in Nebraska. And so what they're all looking at is a very similar thing. It's like, well, what's the makeup of the pathotypes of Phytophthora soji in the fields and within Michigan, South Dakota, Iowa, and Nebraska. So to summarize all 3 tables, I'm going to just use it in this graph, and so I'll sort of spend a little time outlining the graph. But I just selected some of the RPS genes, 1A, 1C, 1K, and 3A. There's a few others that are added within each one of the manuscripts. And then the second column, the percent of cultivars with that particular gene, this is actually published in the Iowa-Nebraska paper, but they're actually using University of Illinois variety testing program data, because every variety that's submitted into that University of Illinois variety testing program, they take notes on the different resistance genes. And so if you look at it, the 1A resistance gene, which was one of the most common years ago, is only in 5% of the varieties that were submitted over the last 10 years. The 1C was the most common, 42% of the varieties. 1K had 17%, and then 3A was at 1.4% that had that particular gene. Now the next 4 columns would be the percentage of the isolates that can actually cause disease on varieties that have that 1A or 1C or 1K or 3A. And so in Iowa, 100% of the isolates that were recovered could cause disease on the 1A. So the 5% of the varieties that had that 1A would not effectively be able to control that particular Phytophthora strain. And in Michigan, Nebraska, and South Dakota, again, over 80% or close to 100% of the isolates that were recovered could cause disease on 1A. So 1C is very similar, and 1K, very similar stories, where at least 70% of the isolates were able to cause disease on the 1C and 1K. The 3A still has some effectiveness, and so only 2% of the isolates in Iowa and Nebraska could cause disease on the 3A, 8% in South Dakota, and 35% in Michigan. So there's a little bit of a bump in that Michigan one, but it's still way lower than the other 3 RPS genes. All right, so here's some of the other nuggets out of these three papers. Is the durability of RPS genes is estimated to be between 8 and 15 years. The RPS 3A is lasting a little bit longer, and you can go back to that previous slide and you can see why. It's not used very much. And so the fact that we're not really putting a lot of selection pressure on that 3A gene means that it is lasting a little bit longer, but it's not being used. One of the other suggestions is maybe to preserve or to steward some of these genes is to stack them, and so that would increase the durability. But if you go back to that same group of variety testing program cultivars that from University of Illinois, only 13 out of the 4,000 cultivars, so 0.3%, had used stacked genes. And so this is not really a strategy that's used very commonly by the seed industry. So in general, the RPS1 locus is not effective, but unfortunately the RPS1C and 1K are the most common genes used. And so this should raise some red flags as far as effectiveness for managing Phytophthora, is that most of the major genes or the single gene resistance that we have out there are not effective against the isolates that currently exist. Another thing that I found interesting is that several of the two or three of the papers talked about pathogen complexity, and this would be the number of resistance genes on which an isolate can cause disease. And over the last 10 to 20 years, this complexity number has increased by 2, which means that an isolate from 10 or 20 years ago, depending on the manuscript, was able to cause, let's say, three or four disease on 3 or 4 of the RPS genes. Now it can cause it on 4 to 6, and so there is an increase in the number of RPS genes that these pathogens can cause disease on. The last thing that wasn't really mentioned a lot in the manuscripts, was sort of in the discussion a little bit, was that partial resistance. This is an option. There's a lot of varieties out there. There's companies that certainly spend a lot of time trying to figure out what the partial resistance is for each variety. But this is difficult to screen for, just to give the companies a little bit of a pass. This is not as clear cut as the RPS genes. And so sometimes these stands or these ratings are based on plant stands, and which means that there's maybe other pathogens that are at play. And so what we found in a couple of instances is that these plant partial resistance ratings aren't always as reliable as we want them to be. So if you do know that you have Phytophthora, there's a couple of options, or here's the sort of the management suggestions, is that use an effective RPS gene, which would probably mean that you're not going to use RPS 1A or 1K or 1C. So 3A would be a good option, but again, there's not a lot of varieties that have that. Select a good cultivar a cultivar that has good partial resistance, and just keep your fingers crossed and hope that that resistance is actually associated with Phytophthora out in that field. And the last one, and we're not going to go into great detail on this, but if you use an effective seed treatment, there's plenty of seed treatments that are out there. If you go to cropprotectionnetwork.org, there are, you can go to the publications, and you can find the publication on fungicide efficacy for controlling soybean seedling diseases, and there's ratings for all of the products there. All right, let's move on to sudden death syndrome. So leg 3 of this talk. Most people are probably familiar with this, but just a couple of slides to catch everyone up if they're not. This is a soil-borne pathogen, so you'll see a lot of foliar symptoms, but this is caused by a root rot at first. Fusarium virguliforme is the pathogen, and it has 2 phases. Again, the first one is the root rot phase, and that happens very soon after planting. And then you have the foliar symptoms, and the foliar symptoms are caused by a toxin that's produced in the roots and translocated up to the leaves. So just to get the thanks out of the way right now, because there's a lot of people and a lot of companies that were involved. But the ones that are underlined are the ones that were part of a USDA grant, where we are looking at trying to figure out if we could find fields that were at higher risk for sudden death syndrome. And so Ryan Hamilton at Michigan State and Edward Ernett here at Iowa State are the two graduate students that are on the project. And then Marty Chilvers and Nathan Klacheski are the two other PIs on that grant. But you can see there's a long list of other people that have contributed to this large data set. All right, integrated management of sudden death syndrome really starts with a resistant variety for resistant to sudden death syndrome, but also resistance to soybean cyst nematode or managing soybean cyst nematode in general. We know that the presence of the nematode can actually increase the risk for SDS. There are also several different cultural practices that are at play that can either increase or decrease the risk for sudden death syndrome. Planting date, plant population might play a role, and different tillage practices. There's certainly a lot of different things that could affect SDS. But then also maintaining or a lot of soil properties, proper pH, different fertility levels. These could play a role in the risk for sudden death syndrome. And then also the use of seed treatments could also be part of an integrated management practice. And this is really what led to this study, this grant that we got with the USDA, where we're looking at SDS risk prediction. And this is coming out of Marty Chilvers' lab at Michigan State, where essentially what he found was that the Fusarium virguliforme abundance in the soil and the soybean cyst nematode eggs, they correlated to the final SDS disease index. And so you could start to predict where SDS was going to show up in the field based on how much inoculum and how many SCN eggs were out in that field. And this actually correlated negatively to the yield as well. And so as you increase FV, or the Fusarium virguliforme, in the soil and the SCN, that decreased the yield. And so this paper was published a couple of years ago, but it led to this idea where we can go out and collect soil samples, extract the DNA, do qPCR, and that would lead to some kind of data analysis, which would lead to risk. Now, the other factor is that there's certainly a lot of field data that could also be collected, and then that would lead to some part of that analysis, which would also lead to helping assess that risk. And this is the part here at Iowa State that Edward Ernett focused on. So the last couple of years, we've had 123 locations in all of the states that were mentioned in that acknowledgement slide a couple of slides ago. So a lot of different states, and each of these states had fields that most of them had a susceptible and resistant variety. Some of them would only have a susceptible variety. All the fields had to have 3 different seed treatments: a base plus a levo, and a base plus saltro. And then we measured foliar symptoms, root rot, and yield when disease showed up, and then gathered a lot of different information. So we collected a lot of soil samples. This led to sending some soil to Michigan State to measure the FV levels, like in the couple slides ago. The SCN egg counts were done here at Iowa State University. Soil analysis and nutrient testing. Soil samples were sent to Midwest Labs. We also did some soil property quantifications using Pattern Ag, and so a newer company out of California, and then also collected weather data. And so a lot of different data that went into it. Just to show you the distribution, this would be the 64 locations in 2020, and then the 59 locations in 2021. So there's a couple of them in Delaware, but in general, most of them are here in the Midwest. Now, some of the fields had an FDX, so this would be foliar disease index greater than 10. And just to let you know, the foliar disease index, that's going to be a combination of the incidence and severity put to a 0 to 100 scale. From previous research, we found that once you get over 10, you start to see a more consistent yield response or yield reduction from SDS. And then, so that you can see, again, mostly in the Midwest, and then the 29 locations that we had that had SCN greater than 2,000 eggs, again, mostly here in the Midwest and then also Delaware. So we're not going to go into a lot of detail because there's still a lot of data analyses that are being done. But in general, we found the top 12 predictors for SDS are listed here. And we're not going to go into great detail, but I'll stay on the slide for a little bit so you guys can read it. But in general, soil type and soil pH were up there. And there's things in here that are not a surprise. The SDS resistance, irrigation. So if you irrigated, you had more SDS. Soybean cyst nematode shows up in here. Seed treatments show up in here. So these are the things that we expected. And then also precipitation at different times of the year is also important. And so we're starting to build up evidence that these are the factors that could be playing a role in increasing the risk for SDS. Now, if we combine that with the level of FV in the field, now hopefully we can get to a spot where we're going to say, hey, this field might be at higher risk for SDS. So, and a lot of the FV data is still being collected from Michigan State and are still being quantified at Michigan State. And so we're just going to focus on the yield responses to the different seed treatments and we'll sort of break it down in different ways. But first of all, all 123 locations, if you look at the yield, both the Levo and Saltro increased yield on average, very low, but is still significant, 1.4% and 1.9% for Levo and Saltro. And if you look at the FDX, the FDX is the orange bar. And you can see that on average across all 123 locations, the FDX was around 7 for the base and was reduced by about 35 to 37% for the two different seed treatments. And these numbers match up pretty well with a lot of our previous data, usually showing reductions between 30 and 45%, just depending on the situation. The root rot was also reduced. And so that was a little bit, it's usually less than the foliar symptoms, but we did see, again, very similar responses to previous studies, but that 12 to 14% reduction for the two products. Now let's focus on the 16 locations that had an FDX greater than 10, and you can see that the yield responses increased to 6.9% for Levo and 8.5% for Saltro. So no significant difference between the two products, but you can see that quite a bit bigger of a yield response compared to the all 123 locations. And then the FDX was reduced now around 46-47%, and root rot was reduced 18 to 19%. And so, again, pretty consistent with fields that have an FDX greater than 10. These numbers match up with previous research quite nicely. All right, so this is one of the slides that's would be new. We have 39 locations that have 2,000 greater than 2,000 SCN eggs. And what you see in this one is that that yield increase now drops. And so first of all, go over and look at the FDX levels. It's back to that less than 10 in the base. And so it's around 7 or 8 in the base. But if you look at the yield increase, so it's pretty much close to 0 for Saltro, and it's a 1.2% increase for Levo. Nothing was significant. The FDX was reduced by 33% for Levo and only 16% by Saltro. And then the root rot was actually not reduced at all either for either product, very close to 0 for both products. And so this is something like whenever I have a graduate student doing research, I always try to figure out well, what's the next step? And it's really based on this slide. I don't know what's going on here, but there's a lot of good research questions that could be raised as far as what's the role that SCN is playing in how well these seed treatments are working sort of in the absence of FDX. Now, if you add FDX back in, so out of these locations, 5 of them had FDX greater than 10 and also greater than 2,000 eggs. And you can see the yield responses go back up to almost 9% for Alivo and 6.5% for Sultro. FDX is being reduced again, that 35 to mid 40s range. And then root rot is also being reduced. And this is only at 3 locations. But again, the data makes a little bit more sense when SDS is added back in. But when it's not, sort of what's going on. And so something to keep an eye on, or sort of future research is there. All right, to summarize sudden dust syndrome, remember, SDS has 2 phases, root rot and foliar. They both contribute to yield loss, and they both should be managed. The foliar symptoms are usually the ones that we're focused on because they're probably contributing more to that yield loss. We know that effective seed treatments can reduce both root rot and foliar symptoms. That's the good news. But finding the fields and the right conditions for to make these seed treatments be the most effective and get your return on investment is really needed. And hopefully some of this research can start pointing into the direction into finding fields that have that higher risk for SDS, where we can get more of a response with these seed treatments. All right, we're going to end with product evaluation of both foliar fungicides, and then we're going to actually go back into the SDS realm and look at the recent data on seed treatments for SDS. So remember, when we're looking at foliar fungicides here in Iowa, the ones that we're really focused on, the diseases we're focused on, frogeye leaf spot, maybe brown spot, which is usually in the lower canopy, and Cercospora leaf blight comes in pretty late in the season. But really the main one would be frogeye leaf spot. So I'll start with regional. And so this was a study that was done in South Dakota, Southern Illinois, Michigan, Purdue, Wisconsin, and here at Iowa State. So 7 locations. And we're looking at sort of a set protocol of fungicides, either in different classes or at different rates, or maybe like one of them, chlorothalonil and tebuconazole, would be sort of a generic, sort of different price ranges as well. And so what we found, and this is just summarizing all of the average across all 7 locations, but what we found is that very little differences in frogeye leaf spot or in Septoria brown spot. You see that if there's anything interesting in here, if you look at the quadrus right in the middle, it looks like it did not affect Septoria brown spot at all. We know that we have resistance to Septoria glycines, the pathogen that causes brown spot. We found it here in Iowa, and it's been confirmed in several other states as well. But it might be a little bit more widespread than we think if we see it averaged across these 7 locations like that. Otherwise, there's just really low disease. All of the frogeye leaf spot is less than 2% in all of the treatments across these states. And if you look at the yield responses, very little yield response in all 7 states. And so fairly boring data, I guess, from the plant pathologist perspective. So if we dive into just the Iowa data, again, this is the 7 locations are the same as the last 10 plus years, where we have 3 research farms in the north, 3 research farms in the south, and then the Ames one in the middle. And we looked at some of the same products, but a longer list of foliar fungicides that are available. And if you look at the frogeye leaf spot across the 7 locations here in Iowa, it's very similar to the 7 locations in the Midwest, where everything is less than 2%. We did see some reductions, but again, nothing really to get too excited about. If you look at the yield responses, the non-treated control had a 64.5 bushel per acre yield. And if you look, there's a couple in there that had close to maybe 2 bushel increases, but nothing consistent or significant across the 7 locations. Now look at sudden dust syndrome. The product evaluation, we had 9 sites in 2021 and 9 sites in 2022. And so this is going to be a little bit different than that risk study. I didn't mention this in the risk study, but that study, we didn't want all of the fields to have as much SDS as possible. We wanted some fields to actually have no risk for SDS or low risk, and then some to have a high. And so in this particular study, all of our fields are either irrigated, inoculated. We try to get as much disease as possible in these fields. So in this one, just the treatments, we had a longer list of treatments, but some of these products were numbered, and so we're not able to show some of that data. But the ones that are available, we have Ceramax, Ilivo, Saltro. I'm going to just call it the AWBA treatment, where we have a combination of several different products, and then a base and an untreated control. There's a lot of numbers here, but 2021, again, 9 locations, non-treated control, the FDX, so that 0 to 100 scale, averaged less than 10, so 5.6. And if you look at the base and the base plus Ceramax and the AWBA one, there was no reductions. Ilivo and Saltro reduced the amount of FDX. And again, this is consistent with what we've seen in the past, is that these are the 2 products that work the best or the most effective at reducing foliar symptoms. If you look at 2022, similar, not as dramatic of a reduction, but very similar responses where Ilivo and Saltro reduced disease, and the Ceramax and the AWBA combination did not really affect FDX. We did have 3 locations that had greater than FDX, greater than 10. And in these 3 locations, you can see, again, similar trend. Ilivo and Saltro reduced FDX, and the other ones did not. Now, the interesting thing for me is if you start looking at yield responses, in 2022, there was really not a lot. Ilivo and Saltro had a couple of bushel increases compared to the non-treated control or the base. And the Ceramax did not, but the AWBA one had a couple of bushel, or at least 1 bushel increase. In 2022, a little bit different story, where we had everything was a little bit tighter. The AWBA one and the Ceramax were a lot closer to Ilivo and Saltro. And so, and then if you look at the 3 locations that had FDX greater than 10, Saltro did look a little bit better than Ilivo, and Ceramax and AWBA were right there in the mix as far as having more of a yield response. And so something that we're looking into a little bit more. We're not dismissing these other products just based on the yield response, but they certainly were not affecting the FDX like we wanted them to. And so sort of a mixed message that we're getting with a couple of these products. All right, so if you want more information, I did mention the Crop Protection Network, and you saw that logo throughout on some of the slides. This is just where we put a lot of our multi-state information into one place, and that's called Crop Protection Network. And so it's cropprotectionnetwork.org. Check it out. There's publications and there's different information available there. And with that, just a huge thank you for listening in on Crops TV. Thank you.